pancreatic lipases that I've mentioned that digest all the remaining dietary triglycerides, what do they require for full activity? Well, they require four things. They require fatty acids, they require bile salts, they require something called colipase, which I'm going to talk about in a second, and they require an alkaline environment. So let's see why they require these four things. Well, fatty acids stimulate duodenal S cells to release, remember fat stimulates S cells to release secretin, and secretin stimulates duodenal pancreatic duct cells, and duct cells secrete a bicarbonate and water secretion to modify the secretions of the pancreatic acinar cells. So this is alkaline. And the alkaline environment is required for full lipase activity. Now fats stimulate duodenal I cells also, and I cells, as we saw up there, they secrete cholecystokinin. Cholecystokinin is the main stimulant for pancreatic acinar cells, and the acinar cells, they're the ones that produce the zymogens, lipases, amylases, and so they secrete the lipases for full lipase activity. The bile salts, as we know, they allow the emulsification of fats by forming bile salt fat micelles. And colipase is a coenzyme that allows ligand binding for full lipase activity. So colipase, let's have a closer look at that. So what's the function of colipase? Well, colipase is secreted along with the other proenzymes. This is a pro-coenzyme, I guess. So it's procolipase, secreted from the pancreatic acinar cells. It's activated by trypsin. Now, if you remember, trypsinogen is a zymogen secreted by the pancreatic acinar cells. It's activated by an enzyme called enterokinase on the brush border of the small intestine to become trypsin, and trypsin activates all the other zymogens. So procolipase is activated by trypsin in the duodenum. It then becomes colipase, and what colipase does is it's like an anchor. It forms a colipase pancreatic lipase complex anchor. So it allows the lipases to absorb onto the emulsion and digest the lipids. So this bile salt emulsion, like a blob of lipids with bile salts on the outside, the colipase is the anchor, it's the link between the lipase enzyme and the bile salt coating to allow the pancreatic lipase, to go in and digest the lipids on the surface. So the lipase enzyme secreted by the pancreas, how does it work? Well, lipase hydrolyzes the first and third ester bond. So the triglyceride becomes monoacyl glyceride and 2 fatty acids. Remember, it's glycerol with 3 fatty acid chains, so if it gets rid of the first and the third, you're left with a monoacyl glyceride and the 2 fatty acids that have come off. And we can compare this to the gastric and lingual lipases because they hydrolyzed only one of the ester bonds, making diglyceride and 1 fatty acid. And there's also other pancreatic fat enzymes, so there's 2 others, pancreatic fat enzymes, apart from pancreatic lipase. There's phospholipase A2, which is PLA2. Now that's active in alkaline pH and in bile salts, and that digests phospholipids. So phospholipids are digested by phospholipase A2, and it digests them to something called lysophospholipids and a fatty acid. Carboxyl ester hydrolase is another pancreatic enzyme, and that deals with cholesterol esters, so esterified cholesterol, and that becomes cholesterol plus glycerol. So the triglycerides have been dealt with the lipase, the phospholipids have been dealt with PLA2, and the cholesterol esters have been dealt with by carboxyl ester hydrolase. And there are also bacterial lipases, so let's have a look at that. Well, bacterial lipases, these work at a high pH. They're not inhibited by bile acids, and they don't require cofactors. Remember, the lipase requires colipase, whereas the bacterial lipases, they don't require that. And what the bacterial lipases do is, because they're very, very effective due to these reasons, it results in the total hydrolysis of triglycerides and phospholipids, and therefore intact glycerides are rarely found in stool. And I guess as a side note, fecal fat is found as soaps in the stool. And just to summarize the various enzymes required involved in fat digestion, you've got lipases, which you've got bacterial lipases, you've got pancreatic lipases, and I guess you've also got lingual and gastric lipases. You have PLA2, which deals with phospholipids, and also carboxyl ester hydrolase, which deals with the cholesterol esters.